I mean, the recording is on. Yeah,
Sir,
go ahead. Go ahead. Yes, please.
the
Go
question
ahead.
is that, sir, if this was the case that we have to attend the classes with our seniors, then why it was not already informed us? Then we would not have missed that. Like yesterday, I have the free time at that time, and I was not doing anything for that matter. So why it was not informed earlier? It should have been informed earlier. Yes.
Who will inform you? It is your responsibility, isn't it? You have to know that you are doing a practical course. You know that there are BBL 433 and this is something which I told your class representative, that the practical component of the course will be done by both you and your seniors. By definition, your seniors cannot be part of BBL 433 or this course because they have already done the lectures. So they will always be part of a practical component of the course. Correct? So think about it logically. Instead of asking for rights, instead of asking for rights, you should also think about some amount of common sense should have been there, and you should have asked why we are not members of 433B. So when I opened it, I discovered that you are not members. This is an initiative which should have been taken by your class. I am now taking it in spite of the fact that my familiarity with IIT system and MS Teams is poorer than
I have emailed many a times to HOD and he did not respond at any of my emails. I have emailed to HOD and HOD office. No response has been given by them. So what should I do then? What can I do in that case?
You should have possibly written to me, who is the course in charge, because instead of getting aggressive about your rights, sometimes you should think in terms of the ways to solve problems rather than saying that I have lodged a complaint to the appropriate authorities. This is not something which is a mountain that is falling on your head and you have to immediately react. You have to have a certain amount of maturity and way ensuring work getting done rather than being righteous about it. So righteousness is very useful property, but righteousness is about property where you don't take responsibility and hope that somebody else should have taken responsibility on your behalf. So that is where, that is the way in which things don't get done, but you all feel very great that I have made the appropriate complaint. So that is why I'm saying that you have only missed one lecture, which is already recorded. So at least your colleague has taken positive action instead of negative action of complaining. So you have to take positive action, and you should ensure that. Now, I do not know why people want to join. I hope they are able to join on their own now. I see requests coming in. Just hold on. My problem with me is that I am also equally unfamiliar with MS Teams and the features thereof. And I would assume as young people, you would have much more familiarity with these things. Now, Vivek, Sahara and 5 others requesting to join team. Okay. I hope.
Sir, I just shared the 433B Teams link with the class. I got it from.
That is it. Yeah. So if you, so all of you, please, if you have the BBL 433 link, be linked with, then all of you can simply join. Is that all right now? Okay.
Yes, sir. That will be very, very fine.
Yeah, and Ratnesh, I'll send you this. Sir, sir, do we also need to have access to the Impartus channel for BBL 433B, or is the Teams?
I personally, you are talking to a person whose knowledge about these things is much lower than yours. So I have never used Impartus. I am told it is a very convenient thing, but I use Teams throughout. Okay.
And nothing on Moodle.
I use the Moodle as well. So you can upload your assignments, your exams, your quizzes will all be on Moodle.
So then I have to ask Preeti ma'am to do us because we can't like the system will have to add us to the Moodle sort of.
Yes, the system has to add you to the Moodle BBL 433B because when the exams and the practical notebook has to be prepared, you people will have to actually be clear that you upload all these on the Moodle. Okay?
Yes, sir.
Okay. Okay. I hope everybody is added and nobody is waiting. Because I do not know why people have to be, why people are seeking invite. I thought they are all part of the, there are so many people asking. Yeah, they are all accepted. Fine, that's all right. Okay. This is also something odd, but anyway. So recording has started. So let us now begin the class. Unless you have any specific questions, I think I keep on getting these messages all the time. We have accepted the request, so it's all right. Why does it keep appearing then? Okay. Okay. So any further questions that you have? So all of you are doing the course online, the practical component as well, and you are now all got the link to BBL 433B, which will be happening from Monday 2 o'clock to 5 o'clock. Other than this very basic introductory lecture that has happened, nothing great has been missed, fortunately. And I would suggest that I'm impressed by your enthusiasm and your feeling of anger that you have missed one class. I really hope that that same enthusiasm is there in attending all classes, because I will be honest with you and rather harsh and blunt that many of your colleagues tend to not attend classes, and a fairly large fraction of your colleagues, a very large fraction of your colleagues do not attend a single class, and then they expect that somehow they will be able to pass the exam by mechanisms which you are well familiar with and which are not something which is acceptable. So you people have all signed the honor code, and yet I see answers, etc., to exams being so similar as to be embarrassing. You know, wrong answers, and as I tell your colleagues, and this is not funny, though I'm trying to make it look funny, wrong answers, and because I asked numericals, and wrong and matching to the third decimal place. Wrong to the third decimal place matching, I used to tell them that at least you should have rounded it off when you were copying, you know, so because it is mechanical and blind copying from colleagues. So these are things which are, and I mean, we have been students once upon a time, long time ago. We can make out who all are using unfair means. Now, is something which we dislike, but we also dislike penalizing. But the IIT has now taken a fairly strong viewpoint on this, and it is not happy with the fact that students are adopting such practices, and which is becoming more endemic as time goes on and you people are doing online classes only. It is time you people became more serious. And I hope that, that, as I said, the seriousness should be to do with actual studies. So we will start the class. I will use the dashboard. So I'll let us start sharing the screen. I think that is the simplest. Tell me, can you see all the whiteboard here? I mean, I should sort of I have been teaching something else here. This is yesterday's class, so. Okay. Can you see the whiteboard? All of you?
Yes, sir. Yes.
Yes, sir.
Yeah. Yeah. So now, as I said, a lot of it depends on your feedback, but that feedback has to be done with sincerity. Let us start with the obvious feedback. What kind of gradation do you want? Now, I would suggest, and you can give your suggestions, but once it is done, it is fixed. There are 100 marks in total, and practicals will be graded independently, and I will talk about the practical grades later on. But for the lecture part, and then of course they will be combined later on for the 4-credit course. But for the lecture part, let us work backwards. We have the major exam, and you know that major exam cannot be more than 40. So the major exams, let it be 40%. Okay, and the minor exam by definition, or the mid-sem can be, let us say, half of it, 20 marks, which leaves you with 40 marks pending. Now, I think that it is a good idea for you people to do some amount of self-exploratory work, though, and not do a very mechanical job of it. And I always think that that's a good idea. So I think there should be a term paper which should carry some marks, where all subjects in biotechnology, by definition, whatever we teach you, is very basic, because the subject is so vast that really I can't teach you in depth. If I start teaching you one subject in depth, I will not be able to teach anything at all. So almost all courses, and this is an important point that you should remember as BTech students and sometimes wanting to be serious about academics, that if you were to be serious about academics, then these courses have possibly the highest depth in any subject whatsoever. So if you start exploring any one subject, you will find that the amount of work done in them is tremendously large. And what we have done is, and you start asking very fundamental questions. So I hope that you do some exploratory academics, and therefore I want to put it as 20 marks, and I want you to pick up any subject of the many in enzyme science and engineering. I mean, as I said, lifetimes have been spent in just one aspect of enzymes. And do a term paper on 20 marks. It's an absolutely open-ended exercise. Anything to do with enzymes, engineering, science, structure, purification, catalysts, mechanisms, whatever you want to do it, just do some exploratory work. It may just end up giving you a feel of how good academics is done. And that's an important component of any, I mean, self-motivation is possibly one of the, and that still leaves us with 20 marks. And so, quiz, let us say 1 and 2. So 10 each, right? So 10 into 2. So that is my 40, 60, 80 and 100. Any suggestions on how to change this, or do we hold this? I mean, I have no problems if you want to change it, but when you change it, think about it carefully. For example, somebody might turn around and say, let us have a 40 marks term paper and we will really do a very serious job of it. Will you? I have no problems if you want to do a very serious job of it. And then I will really go through the term paper very carefully and see how much time and depth you have put into it. Or somebody might say, let's have term paper of 5 marks because we don't even look at things. And so on and so forth. So you can change these numbers, or you can add other structures, or you can remove any one of these. I have no problem. But once you do it, this class determines and finalizes this stuff. So you decide how you want to be graded. Is this okay? Any
yes,
feedback?
sir. Sir, I have a question. Like, what will be the platform for quizzes? Like, will it be only MCQs, or I think it should be subjective questions?
Subjective question means describe in, my friend had this question paper, you know that? Describe in detail and discuss in brief. A, anything. B, something. D, everything. D, nothing. That's a very difficult task, describing nothing. So no describe in detail and discuss in brief. I think I have become a very unpopular teacher, not only with your BTech, but also with your MTech students, because I only give numerical questions. So essentially, I will teach you a lot, but most of the time, if you want, I can give a combination of conceptual questions. I have been giving numerical questions all through. These are all design problems. You are engineers. So it cannot be describe a fermenter, or it cannot be describe an enzyme column, describe how an enzyme works. It is tell me what is the column size and what is the flow rate that I will use for an enzyme column if I want a percentage conversion of 90% to take place over 2 weeks with this kind of Becquerel number and this kind of mixing characteristics and this kind of deactivation of enzyme and this kind of mass transfer issues. That is a very hard problem, of course. You cannot solve this question, but a proper engineering problem with a column which has enzymes which has enzymes which are deactivating, which have which are deactivating with time and which are having mass transfer issues, and mass transfer means both. It could be internal mass transfer, external mass transfer issues, Peclet number, mixing, forward mixing, backward mixing, inhibition, reversibility, and tell me how will you design this column? Now that is a real problem. That is a real engineering problem. You take that one problem, sit on it for 4 hours, solve it and give me an answer. That is the way engineering questions used to be once upon a time. And that is really the way it should be taught. I am told over a period of time that because of this online business, this is not possible. I mean, we were given one problem for 3 hours with an open book and told get the answer to us, back to us. That is it, job done. It's another point that we could never solve it, but I could understand the perspective of the teacher that you are given an industrial problem to solve. So now we actually break up this kind of industrial problem, the larger industrial problem, into small components of problems. Okay, tell me what is the length of the column, tell me what should be the flow rate, tell me how much enzyme should I pack, tell me how do I find out enzyme deactivation rate, and so on and so forth. So usually I mean your previous batch, they were given only numericals. Yes, so sir. The question, the problem with numericals is that when you look at the question, you do not know whether you know the answer or not. You work through it. So it sometimes becomes hard. It's like in your class 12 maths, you know, when you were differentiation was obvious, but when you're given an integration problem, you did not know whether you would know the answer or not. Right? You would sort of start substitution and you do integration by parts, and one or the other would click. So it's like that. So you have to think through your problem and think how to apply principles to a real problem. Yeah, go ahead.
Sir, yeah. So these numerical problems, are they like you give us a question and then we have to fill in a numerical answer in the box, or do we have to write the entire working of how we got to the answer?
You have to write the entire working. I yes, just say here's the problem.
Yes, sir, that is I think what he meant by subjective. Like.
Yeah, if subjective questions are fine. I mean, it's not. I don't believe in MCQs. Because you know you can arrive at the right answer by various mechanisms, including by a 25% probability, right? So I don't, and sometimes you can arrive at the right answer by knowing that the others are the wrong answers. So options are checked. So you know you arrive at right answers by a mechanism which need not necessarily be based on knowledge.
Yes, sir.
So I will essentially try to ask design problems. It'll all be design and equipment to function in a certain way as engineers. And that design by definition is at its crudest would be what is the length, what is the diameter, what is the flow rate. Okay. Yes. I mean, to achieve a certain percentage conversion or so on and so forth. You know, I mean, at the simple, or what is the enzyme loading? Or what is the kinetics, or how do we derive a certain kinetic constant rate, and so on and so forth. So earlier it was only design, but I think now I will not sort of stick to only design. We will ask you some conceptual questions also. Is that all right?
Yes, sir.
Yes, sir. It is all right. But in that case, when you are asking numerical questions, you have to give different questions to different people, because in that case, people will just change the language and use the same formula and in the same way, and they will arrive at the same result. So in that case, I think it's better to give everybody, or at least you have to divide the class into 4 or 5 different groups and give every group a different question. Yeah.
Mr. Ratnesh, right? I think you are sorry. So can you? I mean, I'm sorry, I can't see your face, and you have made a very interesting point that because of the fraud going on by your own colleagues, instead of stopping that fraud, you want me to do extra work. And then when I give different questions to different people, you will say, my group got the more difficult question and that other group was actually easier. So you should now give me higher marks. And this will now, then third, somebody will say, no, our group was more problematic. And why did you make 4 groups? There was cheating within that group. And so why don't you make 75 questions into 4, actually 300 questions. And because, we cannot stop our class from cheating. So for your problems and for problems that should not be there in the first place because you people have signed an honor code, you expect me to take corrective action. Now, this seems to be highly immoral. Is it not more correct that you personally don't copy and don't allow others to copy and ensure that every colleague feels like that? Is that not the more moral
Sir,
and the
I
correct
can
principle?
do.
Yeah.
Yes, sir. It is correct, but I can do that. But how can I go at the time of
You don't
examination?
have
I'm
to.
sitting in my room.
I don't have to.
Yes, sir.
I don't have to. I am only asking you to do it on your own. And I will ask every student
Yes,
to do
sir.
it on their own. But that is all.
I
If
will
I
do
change,
on my own.
please
Thank you.
please don't expect me to make 75 question papers and then the students turn around and say, well, my question was much more difficult than his because you know that trick in that question was much harder to solve than the other fellows because I could solve that trick but I could not solve mine. I mean, this is opening up a Pandora's box of troubles. Nobody can, no examination. Now somebody will say IAS examinations, a lot of cheating is going on. All the 1 lakh students who give should all be given different question papers. No, right? So we will ensure that cheating is not done, but you have to all solve the same question paper. And there are ways to find out, including online, and when you give the exam, you will be under supervision to see that you do not use any unfair means. So let us not get into it.
Yes, sir.
Okay, anything else?
Yes, sir.
Sir, I had a suggestion that can you put 3 quizzes and take 2 best out of 3?
Wow.
Because
that is fine.
Matlab two. Sorry, your name please.
Deepak.
दीपक, मतलब आप हिंदी में मैं बोलूं? दीपक तो हिंदी समझ सकते हैं, हाँ?
Yes, sir.
मुझे लगता है कि 2 बेस्ट आउट ऑफ 3 मतलब जो मैंने मिस कर दिया है, यानी कि मैं छुट्टी मार रहा था.
No, sir. No, sir.
No?
It is sometimes when we are preparing for the exams, at the beginning we don't have quite a good idea that how would the questions were asked and how.
Okay. If you want 10 into best of 3, please remember.
Yes, sir.
Okay, I will make 2 remarks here. These quizzes will be surprise. Then these will be surprise quizzes, not announced. They will take place during class time. During class, I will suddenly say, well, today I have a quiz, and there will be 10 minutes, 1 question only. So they will be as much a measure of your ability to solve the question as well as your ability, as your attendance in class and your interest in class. Usually it will be from the previous class. Is that all right?
So this is for best 2 out of 3 or?
Best out of 3, 2 out of 3.
No, sir, then I guess like a non-surprise quiz would be better. 2 non-surprise quizzes would be better. At
So
least
now
that
you decide.
But like that's what.
So you tell me which one you want. I can take 2 best out of 3, but then there will be surprise quizzes and there will be 10 minutes, 1 question. So there will also be a measure, an indirect measure of class attendance and interest and showing interest, regular interest in class.
Yes, sir. Everyone's saying please no surprise quizzes. So yeah, if you could just do.
Why? What happened? Bad, huh? Bad of me.
So, like, even if someone misses a class for some genuine reason for 1 day, then that they might be in very.
Yeah, but that is why it is 2 out of 3.
Yes, sir, yes, sir. But still, yeah, and like everyone's saying this same thing.
Okay, tell me tomorrow.
Yes,
it will be 20 marks for quizzes. How you want them? 4 quizzes, 5 marks each, but it will be just 1 question during the class, whatever.
Yes.
But then if I see that the class attendance is only max when there is a quiz and other days it is not, we will shift to surprise quizzes back again. I'm sorry, I will keep this option open. If I have had, what is your class strength right now?
Sir, I think it's about 50 to 60 people.
Right. And if I end up getting a student, the day I have a class strength of, let us say, less than 15, I'm going to have a quiz. That makes me easy for correcting answer scripts.
Oh, yes,
sir. So if I see non-seriousness in your class, then we'll have to handle this appropriately. Anything else about coursework? Okay, I will upload the books, whichever don't have all the books with me right now. So I'll upload the books, at least 2 of them on enzyme science. Enzyme engineering books are not easy to get around, but enzyme science is what we'll focus on. We will mostly focus on, I think the course will be essentially enzyme kinetics. Now let me discuss something about the course. Enzyme kinetics is the main thrust area of the course. Now, before I I want to ask you, and then we will decide. Enzyme kinetics and it's associated with enzyme engineering. When you add mass transfer and stuff like that, it becomes enzyme engineering. So enzyme kinetics is the main thrust of the course. Now, how much are you familiar with enzyme production, recombinant protein? I think there is an overlap with other courses and I want a feedback from your side, and then depending on that I can start teaching that as well. For example, are you familiar with recombinant techniques for overproduction of enzymes?
Sir, we have not learned in any course about this, but like we have learned a few things from like on our own online. So that may not be the case to everyone.
So you want me to talk about enzyme production as well? You have not done anything on enzyme production?
No, sir, not like we have
Especially
done.
Because enzyme production is there are 2 aspects to it. One is the boring aspect, which is the enzyme production using fungal cultures, which is more like industrial microbiology. And I really don't want to teach enzyme production using industrial microbiology. This is something which really has no insights. It's not that I don't want to teach it, but it is a descriptive sort of boring topic. But recombinant enzyme production, have you done recombinant enzymes? No.
No, sir. Sir, actually, we are doing a course on industrial microbiology this semester, BBL 431 under Professor Zia. So, but we haven't had a class, so I guess we.
But I think that that course is mostly much simpler, isn't it? It touches on the surface, skims the surface of multiple steps in industrial microbiology, right?
Sure.
So,
and I don't think it talks about enzyme production. It talks about the production of chemicals, other basic chemicals.
Okay, so let us begin with the recombinant enzyme production. Let us forget enzyme kinetics. That's what I taught your previous group, and we went around for enzyme kinetics for very long. And at some point, I think the students were unhappy because we got into too much of depth, and that kind of depth is not really required. I later on felt that very complex details of enzyme kinetics are not really important. They get mathematically very confusing and they become very tedious mathematically. So let us not do that. So let us then spend the first couple of months, or what am I saying? 4 months is the course. Let us spend the first month on recombinant enzyme production, teaching recombinant enzyme production. Is that all right? And here it will be more, as you rightly said, this part of the course will really not have any design issues, but let us begin with this. Shall we begin then with recombinant enzyme production, or do you want me to put it in the back and start with enzyme kinetics? Any suggestions on how you want the course to be structured? Today I just want a feedback on everything so that you're most comfortable.
Sir, can you please repeat that point?
I'm saying, do you want to, this is one topic that you said you have not covered. The main focus of the course is enzyme kinetics. So have you done a course in reaction engineering and chemical engineering?
Yes, sir, we have done a course in reaction engineering.
So essentially enzyme kinetics will build on reaction engineering. So you want me to start here or you want me to start here? Enzyme kinetics? I mean, I just
Yes,
some,
sir, we can
you know,
start with recombinant enzyme production, I guess. I mean,
you
yeah,
want to, okay, enzyme production पे शुरू करते हैं. But you know,
yes.
I want you to be involved, and a part of the involvement, sense of involvement, comes when you say, dictate the course structure. When you say, I want to learn this, I want to learn that. You know, it helps you to own the course. So we will start with the recombinant enzyme production then. Okay, all of you
Yeah.
agreed? So shall we begin?
So,
Yeah,
I mean,
already
no
half.
one has said anything about it. Like, if other people can pitch in, I mean.
हाँ बताइए आप लोग. हाँ, it's open. Yeah, I know. Please, any opinions? I mean, or you think it's either which it's all right. So ठीक है, let us do this, and you guys need to stop me and say, sir, ये एक महीना हो गया है, अब ये enzyme kinetics पे आ जाइए, because this is a much larger course. This is a much larger course than this. Of course, both are very large, so we have to give proportionally sort of this. This should be 1 month and this should be 3 months, something like that. Okay. So shall we begin with enzyme?
You only have a total of 3 months.
Right, sorry. So then we are already lost one class. Let us quickly begin. Let us not have any further discussions. Enzyme, recombinant enzyme production. Okay?
Yes, sir.
So, and obviously, because we are going to talk about simple stuff, let us start the class with the fact that we will work with the workhorse of recombinant enzymes, and the workhorse of recombinant enzymes, and this is the course that you have done, is E. coli. There are, by definition, enzymes are the main product, and you are all familiar with enzymes, right? I don't have to get into what are enzymes and stuff like that. They're biological catalysts, as far as we are concerned, and they are, and you have done a course called recombinant DNA technology, right? rDNA.
No, sir, we have not done.
Oh, you have not yet done rDNA technology?
No, sir.
So that is the next semester.
That's the next semester. So then recombinant enzyme production might become, but you're familiar with plasmids and vectors and stuff like that?
Yes, sir. Somewhat.
Yes,
And you have done, okay, so I mean, instead of asking questions as I'm doing now, if while teaching in the class, if you think that this is something which I have jumped or I have not given enough background information, then you should stop me. You have done a course in molecular biology and biochemistry, right? That's
Yes,
what
sir.
you have done. So one of the things that E. coli, E. coli, as you know, is a prokaryotic system. Now, if it is a prokaryote, prokaryotes have no post-translational modifications. Now, what is post-translational modification? Post-translational modifications. Now, you have the DNA making the RNA, mRNA actually, but I'm just writing RNA. Now, this is called the transcriptional step. Transcriptional step, and then the RNA becomes the peptide, polypeptide. This is all, and this is called translation, right? And then the polypeptide folds to the protein of interest, and then, of course, after folding, it could undergo export to the extracellular medium or to the periplasmic space, either to the periplasm or remain intracellular or get exported. I mean, right outside. Usually gram-negative strains like E. coli, export is usually a question mark. It usually reaches the periplasm. You are all familiar with terms like periplasm and so on and so forth, right?
Yes.
Yeah. So now at this point, the protein can actually have undergo post-translation changes and folding and post-translation modifications, which do not take place in prokaryotes. Usually post-translation modifications are in the form of glycosylations, usually glycosylations, mannose, etc., etc. You know, I mean, I'm not going to get into details of these glycosylation modifications because this is a very important component of giving protein its structure and function. Now, so a protein, and I will draw a very funny graph here, and I can say, let us say complexity of the protein. I have a very complex protein or complexity of the protein, and I have the complexity of the host. Now, complexity of the host means that the E. coli here is very, very non-complex. This is E. coli. So if the protein is not complex, you can use E. coli to produce the recombinant protein of interest. So this is here is E. coli sitting at the bottom. Very useful, very quick production mechanisms, very easy to grow, very easy to produce, and we will talk about all those things later on. But it can produce proteins which are simple, which do not have post-translation modifications, which have simple folding patterns. On the other hand, something like yeasts occupy the middle ground. Yeast cultures are eukaryotic cultures, and yeasts usually you have heard of Saccharomyces.
Yes, sir.
Cerevisiae, but nowadays this was the first preferred yeast for production of recombinant proteins, but now much more preferred is the methylotropic yeast because they methylotropic yeasts. Now methylotropic yeast mostly the Hanesuella polymorpha or Pichia pastoris. They are very similar. Pichia pastoris is the most common name. Hanesuella polymorpha is a variant of methylotropic yeast, slightly different from Pichia. This has become more popular, primarily because it is sold by Invitrogen. So yeasts occupy the middle ground because they are eukaryotic, but they can be grown much more efficiently. Then, of course, come things that we will not talk about, things like baculovirus cultures and insect cell cultures, baculovirus waste production mechanisms, insect cell cultures, and so you can get more complex proteins. And then the most common of all these, and nowadays the most preferred, is what are called mammalian cell cultures. Now, mammalian cell cultures are used for almost all mammalian cultures, and usually they are called, for example, the most common is, I think this is a very bad color to use, Chinese hamster ovary, CHO cell lines, or, you know, and this is what you learn in your mammalian culture systems. CHO cells is Chinese hamster ovary, various, I am trying to give you a very brief overview of how production systems look in recombinant enzymes. All these are producing enzymes, and the enzyme is becoming more and more complex. CHO cell lines are used for almost all human therapeutics. And I do not know whether you are appropriately aware or impressed by the growing market of human therapeutics. One single invention of a monoclonal antibody, MAB as in short, a monoclonal antibody and produced in CHO cell line, one fetches you about $5 to $10 billion.
Sir?
Yes.
I had a question like, what are cell lines? I'm not knowing about that.
Okay. These are cell lines, meaning these are different kinds of cells. E. coli is a prokaryotic cell. Okay. You're familiar with E. coli? It is a bacterial cell. It is a prokaryote. Yeast is an eukaryote, but it is a single-celled eukaryote. It can be grown in fermenters.
Yes.
Okay. Sir. Insect cell lines you would not have handled, but insect cell cultures can be grown and then infected with baculovirus, and which is used to produce proteins. Each of these is modified by recombinant methods to help them produce enzymes or proteins. These proteins, mostly enzymes, are used for various purposes, both for in terms of catalytic activity, but also for mostly, as I said, human therapeutics. Enzymes as human therapeutics, or for assays, or for what do you call some kind of vaccination, or for diagnostics, okay, or for assay of any reaction that will help you identify some disease, or industrial diagnostics, whatever you want to. So a range of enzymes are produced by these cells. Mammalian cells are cells derived from mammalian systems. So they are grown in fermenters, but they look quite different. Have you come to the department and seen mammalian cell cultures being grown in red colored flasks? No. Have you seen E. coli being grown? Have you seen E. coli or yeast being grown?
Yes, sir.
Sir, I have not seen, sir. Sir, like you can think like nobody yet have gone to lab, so how can we have seen?
No, you have not gone to lab for the past. When you joined, there was COVID or what? Okay. By the time you reached the lab department and started working, the COVID protocols had come in, and last 2 years you were shut down. So don't worry.
Sir, we have never gone to department. We were in first year and the COVID came.
This is just bad luck. I know this is really bad luck, but it would be very exciting for you people to see how E. coli is a simple task. This is something many of you would have grown. You can just put it in a flask with medium and it starts growing. So with yeast. Mammalian cultures are cell lines which have to be handled very, very carefully. They're called cell lines for reasons that I will not get into it. They are cells which are not easy to sort of culture and easy to grow, and different cell cultures. There are different mammalian cultures, so there are different names, HEG and DG44 and all these names. Once you, this is the whole, the whole place where biotechnology is happening, mammalian cultures, because each of these mammalian cells appropriately modified could help you produce a human therapeutic protein, a monoclonal antibody to be precise. And I'm talking about these in vague terms deliberately. This is more general knowledge, today's class, and then we will get into the process of production in an engineering sense later on, but you should have an overall perspective that mammalian cultures are used to produce recombinant proteins, monoclonal antibodies, because they have a glycosylation pattern, or the way glucose and mannose is added to the protein, in ways which are very similar to humans, and therefore the only way that that particular drug, MAB, is accepted by the Food and Drug Controlling Authorities as a drug. You can make the same protein in E. coli or Saccharomyces, but your glycosylation patterns will be different. So the protein will be subtly different from the original human protein, and which could cause a reaction. Therefore, the only acceptable way of producing a monoclonal antibody is to have a glycosylation pattern similar to humans, and hence only in mammalian cultures. And therefore, mammalian cultures which produce human therapeutics or monoclonal antibodies, and you can check the names. This is an exercise that I leave for you, and find out what are the, and each one of these, not each of these, each one of these, whoever owns that clone and owns that mammalian cell culture has a $5 to $7 billion turnover every year. So you can well imagine that there is large amounts of money at play for being able to produce enzymes more efficiently. So, and the only reason you move up in complexity is this ability of the cell to carry out subtle modifications in the protein which it is producing, which E. coli cannot and mammalian cells can, the maximum. Yeah, please go ahead.
Sir, my question was that like Food and Drug Controller Agency, like it is in India also, or is it an international organization?
It is an international organization, but it is very much in India. All India, this is biotechnology is a world system. It is, you forget about the fact that if you want to, nobody wants to make a product and sell only in India. If you want to make a product and come to me as a startup and say, sir, I will make this protein and sell in India, I'll say, forget it. You have to sell all over the world. And if you want to sell all over the world, from Baddi to everywhere, all Indian manufacturers, they are regularly checked by FDA and they get FDA clearance before they sell the drug. The DGSR of Indian controller is exactly similar to FDA. So when you get one clearance, you get all of them. So we keep on talking of FDA as the general most common international body. It is a US based body, but for all practical purposes, these are synonymous. And you need an FDA clearance to actually sell your product.
Yes,
but
sir. I did ask this question just because I think that FDA is United States. It is
yes,
not an international.
It is a US body called Food and Drug Administration. But it is the most commonly accepted. It is the most commonly accepted regulatory agency because its rules and methodologies have been over a period of time standardized, and most people tend to copy FDA rules, including our own Director General of Safety (DGSR) clearance, which we get. For FDAs, and because you want to sell it in the US, because the market size is the largest there, normally most industries prefer to get FDA clearance. Anyway, that's part of the story about industry. Really, the point that I'm trying to make to you here is that proteins can be made, the simplest of proteins can be made in E. coli, and then in yeast, baculovirus, mammalian cell cultures, and it is this mammalian cell cultures which produce high-end proteins for human therapeutics. E. coli, low-end proteins, but equally valuable for diagnostics, for industrial microbiology, for industrial biotechnology, and as well as yeast. Yeast also now tends to occupy a very large area. So usually nowadays this has gone out, baculovirus, E. coli, yeast, and so we have recombinant enzyme production methods using different host systems, different host. So these are the hosts. The host is E. coli or yeast or this, and this is the product. So what host you will use will depend on the complexity of your product. How much modifications, what is the complexity of the protein that you're producing, and that determines. So we will talk, I mean, most of my engineering work, that I can talk about mammalian, but that will be wasting too much. Most of the engineering work will be focused on E. coli and yeast only. So any questions? Let us stop here. Tomorrow we will start the basics of recombinant protein production in a more fundamental way. Today was just a very brief, more common sense kind of introduction. A few questions?
Sir, I have one question.
Yes.
Sir, you said that market in United States is bigger. So, sir,
yes.
How can it be like the India's population is like 1.3 billion and United States population is just 330 million? So how can market be bigger there and not here?
आप हिंदी समझते हैं? ये जवाब हिंदी में ही ज्यादा अच्छा लगेगा सुनने में.
हाँ सर, हिंदी तो
वो ये भाई साहब ऐसा है, हमारे देश के लोगों के पास पैसा नहीं है दवाई खरीदने का. ऐसा नहीं कि दवाई की जरूरत नहीं है, उनके पास मार्केट जो है वो कितने दवाइयों का डोज है उस पर ही डिटरमिन होता है. अफ्रीका का मार्केट इतना बड़ा है लेकिन लोग खरीद नहीं पाते हैं. और यूएस में फॉर वेरियस मैकेनिज्म्स इंक्लूडिंग बाय द फार्मा देमसेल्व्स, द प्राइस ऑफ ड्रग्स इन द यूएस, अगर आप यूएस कभी जाएंगे तो आपको समझ में आएगा कि इंडिया में जो आप आराम से खड़े होकर दवाई खरीद लेते हैं ना, यही दवाई आपको 50 गुना ज्यादा पैसा देना पड़ेगा, और वो भी आप खरीद नहीं सकते हैं डॉक्टर के प्रिस्क्रिप्शन के बिना. सो वन ऑफ द बिगेस्ट प्रॉब्लम्स इज बाय वेरियस मैकेनिज्म्स, मोस्ट ऑफ इट टू डू विद issues that I will not discuss, but most of it, the price of drugs in the US is 10 to 50 times higher than in India. So that is why. Yeah. Go on.
Sir, are there any recommended books?
I will try to see whether what now, because we did not think about recombinant enzyme production as part of this course. The enzyme kinetics books I already have. Now I will have to locate the recombinant enzyme production books after this. I mean, you'll have to give me some time. Not by this week, next week only. I will locate some books. I had thought I would start with enzyme kinetics. So enzyme kinetics के books मैंने तैयार कर लिए थे. But unfortunately, recombinant enzyme production, there are many books. But I'll have to choose one or the other and try to get it in online mode and upload it. Mentioning the book to you is not good enough. I mean, I need to get the book available online and uploaded. Let me see. Okay.
Yes. Thank you.
But of course, what I was talking today is something which is there in any good quality biotech general knowledge system. So today it has been an introductory class. Okay, we'll dive deeper later.
Thank you, sir.
Anything else? Okay, thank you.
Thank you, sir.
Thank you, sir.
Okay, we will tomorrow 9 o'clock. Thanks a lot.
Thank you, sir.
Thank you.
Thank you.